Alright, so now that we know the genetics is carried in the sperm and the egg, we have to figure out which one's going to win. We have to figure out why you have your mom's curly hair, or your dad's chin, or why Snape thinks Harry has his mother's eyes when he obviously does not. You can actually figure out which one of the traits is stronger and which one will eventually win out. So the first dude to actually try to do this was Gregor Mendel. He's a teacher at a monastery, and he had a bunch of pea plants, and some of them had round and then wrinkled seeds, and he didn't know why. So he performed experiments where he put them into different patches, and then looked at the parent generation, the pea generation, and then looked at their offspring, F1, and their offspring, F2. And then he compared genetic similarities amongst the little bundles of pea plants that he kind of planted all over the place, and was able to see which traits were passed on and which were not. So we call this Mendelian Genetics. Mendelian Genetics is very straightforward, and it's very black and white. It's not going to cover all genetics, but it's the basic rules of one trait being dominant and one trait being recessive. Dominant means it is more powerful, and it's a stronger trait, and recessive means it's weaker, and it will get overshadowed by the dominant. So in the case of Gregor Mendel's peas, maybe one of them is smooth, and that's dominant, so it's going to be a capital letter. Each letter represents one trait, so we'll just choose an A. And the wrinkled seed is recessive, and we'll use a lowercase a for that one. If you get a parental contribution of A from one parent, uppercase A, and lowercase a from the other parent, the uppercase A, the dominant trait, will win out, and you will not see the recessive. This unit has a lot of vocabulary and a lot of new words, so we have to make sure we understand these words. First of all, this could be the combination of traits that pea plant got from its parents. The combination is called the genotype. This is all of the letters that you have in your genetics. However, as we just explained, I'm only going to see the capital one because it basically overshadows the recessive. So that means smooth is the phenotype if we assumed that capital A was smooth and lowercase a was wrinkled. I'm only going to see my phenotype. So my genotype is the genetic makeup, the big A, the little a, or big A, or little a, and smooth is the physical trait I see, pH. That's what I'm physically going to see. If you have brown hair, you might be a carrier for a recessive trait for blonde hair, but we don't know because we see the physical phenotype. So there are three combinations that you can get from your parents. You're going to get one allele from the sperm and one allele from the egg. So the first combination, getting one dominant, we'll use purple for dominant and red for recessive. One dominant, one recessive. These are different cases. We know from a couple units that the prefix homo means same and hetero means different. So if these are different cases, this is known as heterozygous. The next one is two dominants. They are the same case, so this is homozygous. The third one is two recessives, so this is also homozygous. However, we need a way to differentiate between homozygous with two capitals and homozygous with two lowercase. So we need to add a second word. If you are two dominant traits, you are homozygous dominant. And if you are two recessive traits, you are homozygous recessive. Those are the three different combinations of Mendelian genetics. So since our dominant overshadows our recessive, if you are heterozygous, you get the dominant phenotype, whatever that may be. If you are homozygous dominant, you also get the dominant phenotype because I'm going to see that capital A because there's no other option. However, if I have no dominant capital A, I'm actually going to be able to see the recessive in homozygous recessive. So the first two combos will yield the same phenotype. The third combination will actually have the recessive phenotype. I will see something different in this guy than I do in these two. So Gregor is kind of the father of genetics, and he started something called a Punnett Square. This is how he was able to run his experiments on his pea plants. I'm going to show you monohybrid and dihybrid crosses. Let's do monohybrid first because mono means one. That means we're looking at one trait. Say one pea plant is heterozygous. I'm going to cross it with a pea plant that is homozygous dominant. So what I'm going to do is I'm going to draw a little square right here and basically make what looks like a window with 4 boxes. It doesn't matter where you put the male and the female. I can put the sperm here and here and the egg here and here, or vice versa, as long as one parent is on the top and one parent is on the left. I can't have an egg here and an egg here. So since I didn't tell you which one of these is male and female, we'll just choose one. We'll do this one first. Capital R, lowercase r. Second one, we'll put right here. Big R, big R. So now you have to do what I call battleshipping. When you play Battleship and someone says D5, you're going to look at the column up and down, and then you're going to look at the number left to right. So we're going to look at our columns. Our big R goes down that way. Our little r goes that way. That means our big R on the left goes that way, and a big R on the right goes that way. Any place there's an intersection of lines, you're going to write those 2 letters. Intersection there, 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 and there. So in my first box, I can put my R from the top and my big R from the left because they intersect in that box. As a rule, to keep all this organized, you always want to write the capital letter first. So in the top right box, I have my pink R from the left and my blue recessive R from the top. Bottom left box, blue dominant R from top, pink dominant R from left, which means our last box has to have the pink dominant R on the left and the recessive R from the top. So these 4 boxes are a cross of these 2 parents. That means these are their 4 potential offspring. Let's see how many genotypes there are. We have a homozygous dominant, RR. We have that a second time, so we don't need to write it again. And we have a heterozygous, capital R, little r, and we have it a second time as well. So if I was to give a ratio of the genotypes, there's 2 homozygous dominants and 2 heterozygous. And I can make that as a ratio of 2 to 2, or even reduce it down as 1 to 1. So there's the 1 to 1 ratio. I'm going to get those 2 combinations of genotypes. Let's look at our phenotype ratio. Let's say that the capital dominant R is for a green pea. So anywhere I'm going to see the phenotype green expressed, I'm going to highlight it. Box in the top left, capital R, I'm definitely going to see green because capital R is the only choice. Bottom left, capital R. Top right is heterozygous, but the dominant overshadows the recessive little blue R from my sperm, so I see the phenotype capital R. Same thing in the bottom. So this one's actually really easy. It's the 1 to 0 ratio because there's 100% chance for the phenotype to be expressed with that dominant green trait. So genotype and phenotype ratios can be different. Remember, this is a monohybrid cross because we're looking at one trait. That's why there's 1 letter of the alphabet present. I'm going to do one more example for that one. Got a heterozygous dad crossed with a heterozygous mom. We'll put dad up here and mom over here. We will battleship up the letters and then we'll look at our combinations. Say I want the phenotypic ratio for this cross. Let's also say capital B equals brown hair and lowercase b equals blonde hair. So blonde is the recessive. Box in the top left. 2 capital B, so I got 2 brown, so that's for sure going to be brown phenotype. Top right, 1 capital, 1 lowercase. My capital is a dominant that overshadows my recessive, so still going to see the brown phenotype. Bottom left, we have the same thing. Capital B overshadows lowercase b. And then finally in the bottom right, I have 2 recessive, so I'm actually going to get a blonde there. Ratio's 3:1, or you could even say 75% brown. I have a 75% chance to have a brown-headed kid if I crossed up these 2 people's genotypes. So that's monohybrid cross. However, there are tons of traits, and we want to be able to do a little bit more. We want to get the chance for maybe 2 combined traits. So to do that, we have to do a dihybrid cross. It sounds complicated, it's really not. Say we're going to look at hair color and eye color. I'll use H for hair color and E for eye color. We got a dad who is heterozygous for hair color and heterozygous for eye color. Those are his 2 genotypes for those 2 traits. Cross it with the mom, who is homozygous dominant for hair and homozygous dominant for eye color. And to keep this easy, we'll say that black hair is dominant, blonde is recessive, using this key up here in the right, and brown eyes are dominant and blue eyes are recessive. So a dihybrid cross is just going to be 2 crosses. But I have to put each trait in there, and then I gotta get the calculation to figure out the 2. Got my 2 crosses done. We're gonna compare the hair colors of both. I'll take dad's hair color, mom's hair color, and then dad's eye color and mom's eye color. Battleship time. So there we go. So now, what if I wanted to figure out the percent chance that my kid is heterozygous for both traits? Heterozygous means different. So I'm going to look over on the left and I'm going to say heterozygous for H for hair color. 1, 2 out of 4. So I'm going to give a reduced fraction. I could put 2 out of 4. It's just more math. I'm just going to put 1/2. On the right, heterozygous for eyes, 1, 2. Also 2 out of 4 boxes, or 1/2. So there's a 50% chance that I have either of those things. But I asked for the percent chance that I have both of them. So to do that, I'm going to multiply my 2 fractions. 1/2 times 1/2 equals 1/4, which if we need it in a percent, 25% chance heterozygous for both. Dihybrid is super easy, and all you do is a second monohybrid and then multiply the fraction. If it was 75%, it's 3 out of 4. 50%, you can do 1/2. And if it was 1 out of 4 for 25%, well, you can kind of figure it out. And if it was 25%, it's 1 out of 4. So as with everything in science, there are exceptions. Mendelian genetics was super straightforward. It was, alright, big letter eats little letter, the end. You get the dominant trait. However, some traits are not that cut and dry. Some of them don't follow the rules of get this or get that. They actually do different things. This is called non-Mendelian genetics. I'm going to show you a couple examples of each one. So in this first example, say I have a blue flower and I have a red flower. So let's just use a random letter. So in Mendelian genetics, that capital F would overshadow the little f. However, this is non-Mendelian genetics, and this is the exception. In this case, it's called incomplete dominance. So we would take the F and the little f, and that flower would actually be purple. Incomplete dominance is a fusion of the traits, or kind of a blending. We got our same blue flower, we got a blue color, and a red color trait. When they combine, instead of the Mendelian genetics or the incomplete dominance blending, we're actually going to do something called co-dominance. If you want to think of this like a co-captain, co-captains are the same strength in the team hierarchy. However, they're not going to fuse into one giant captain. They're separate. They are separate things, but they're equally powerful. In the case of co-dominance, one of the traits will be expressed as well as the other. They do not fuse into a purple. They're distinct and I see them. So incomplete dominance, say we have a black dog here and a white dog here, that dog is going to be gray. In co-dominance, if you have a black dog here and a white dog here, maybe that dog is white with black spots, or black with white spots, or a lot of black, a lot of white. You're basically going to see each distinct color. So since those don't follow the normal dominant overshadow recessive rule, these are non-Mendelian genetics. So now we can get into a couple fun ones. The next three non-Mendelians we talk about are polygenic, multiple alleles, and sex-linked. Polygenic means that multiple genes affect the trait. For example, this is like eye color. Eye color isn't always going to be completely dominant, completely recessive. Sometimes you have mixes and matches. So multiple genes out of your DNA are actually going to affect that trait. Multiple alleles means that there's more than 2 alleles per gene. The example we're going to go over is blood types. And then finally, sex-linked means that the alleles actually reside on the 23rd pair of chromosomes. For sex-linked, females are XX, males are XY. That means the allele has to go here, here, or here. There's not going to be an allele on the Y. So those are the last three. And now we can do some examples for blood type, which is one of my favorites. We're not going to worry about positive and negative for the blood types. We're just going to worry about the 4 phenotypes for blood. Type A, type B, type AB, and type O. Type A blood has little bitty A proteins residing on the blood cells. Your body recognizes this, so anything that pops in with a different protein, it's going to kill. Which means it has B antibodies looking for anything with a B protein. Because it knows that's not one of its normal blood cells, it's going to treat it as an intruder. Type B blood has a little B protein on top of it. As you can probably guess, it thinks anything with an A is an intruder, so it has A antibodies. Type AB blood has both, so it's got the A protein on it and it's got the B protein on it. Because of this, it can't have any antibodies at all. If it had A or B antibodies, it would kill off its own blood. One cool thing about AB is since you don't have any antibodies, you can actually receive any blood types. So you're what we call a universal recipient. Last but not least, we have type O. O has no proteins on the blood, which means O, unfortunately, is going to think that anything coming in with A or B is foreign and it's gonna kill it. So you can't get a blood transfusion from any of the other 3. That's also what's cool about the type O blood is that you are actually a universal donor, because your blood without proteins won't get tagged by anybody else's antibodies. You can also only get blood from type O, so people who have type O blood should donate as much as possible because we can never get enough of the universal donor blood. So here are the genotypes for the blood types. Since they're all the same trait, the trait is blood. So we're going to use the same letter of the alphabet for this trait, just like we did on hair or eye. However, we have multiple alleles, so here's how we're going to do this. We're going to use the letter I, and we're going to show the dominant A blood as I to little a. Since we can follow some of the same rules we learned from Mendelian genetics, I know the genotype can be homozygous dominant, and I have type A blood, or it can be heterozygous, which is going to be one of the dominant and one of the recessive. Both of those are going to yield the phenotype of type A blood. We can do the same thing for type B, because type B is actually codominant with type A. I with a little b up here, or heterozygous B with the recessive. Type AB only has one possible genotype. I gotta get one of the A's and one of the B's from the parents. And you'll notice on our blood cell, the proteins do not fuse into something new. They don't fuse into some letter in between A and B. I don't even know what that would be. But you actually see both. So we know that's actually a type of codominance. We're going to see both the A and the B. And then finally, O. O is when you get two of those little recessive I's. Those are genotypes for blood type. They're going to follow the same rules as dominant recessive Mendelian genetics, with the exception of AB, which has a little bit of co-dominance. So because they have some co-dominance and they got the multiple alleles, they're technically classified as non-Mendelian. Here is an example of how that would play out. Say your dad is heterozygous for type A. So we got one of the capitals, one of the lowercase. Cross it up with the mom, who is heterozygous for type B, capital and a lowercase. We'll build it just like we normally would. We put dad up here, we put mom over here, and then we battleship our blood types. If you don't know how I just did this, refer back to the monohybrid portion of the video. So there are our potential children blood types. So let's do a phenotypic ratio. That's going to be our question. I'm going to highlight any phenotype for type A in red. And it looks like this one on the bottom left is going to yield type A because I have one of the A's and one of the recessives. I'm then going to tag type B in blue if we got any. Now look at there, top right. That kid's going to be type B. Heterozygous for type B, but still phenotype is type B. And then I'll use another color, let's say green for type AB. One on the top is going to be type AB. And then the one on the bottom we'll use another color for, oh, little I, little I. So this is actually a cool phenotypic ratio where we are actually going to get 4 very different blood types from a type A heterozygous dad and a type B heterozygous mom. Since we have one of each, it's actually a 1 to 1 to 1 to 1 ratio. And you can write this a couple different ways. You could write 25% for each. You could write 1 to 1 to 1 to 1. You could even do 1AB to 1B to 1A to 1O. You just got to make sure that you know that's not 10 at the end, and that's actually the letter O. Genotypic ratio would have been just as different because I have 4 different genotypes in my blood type. So blood type works the exact same way as a monohybrid. Moving on to the coolest one, sex-linked traits. Remember, these are going to reside on the X chromosome. On that 23rd pair, the XX for female or the XY for male, it's going to reside on the X. It cannot reside on a Y. So we already have a lot of this set up. So since there's no other real options, you got the XY for the male, and the XX for the female, your Punnett Square is going to look like this every single time. This makes sense because that gives us 50% chance. This makes sense because it actually gives us 50% chance for a boy, these 2 possibilities over here, and 50% chance for a girl. It's roughly 50-50. So if you ever hear about techniques to like increase the chance for a boy or a girl, like eat 50 lemons, you know that it's just old wives' tale nonsense. So since we know the allele can only hang out on the X, we're actually going to put it up on the top just like we did with our blood type. So assume we have a dominant capital R, that is the healthy trait. If you have a lowercase recessive r, I'm going to use red for bad, I guess, that means that's the disorder, or that's the disease. Let's use male pattern baldness as an example, and I'm actually going to show you why you can kind of blame your mom if you end up being bald. So say your dad's bald. We're going to put a little lowercase r right there. You can't put it over on the Y. This has to stay blank because the allele only goes on the X chromosome. So say your mom has a full head of hair, no baldness. So she's got to have at least one of the dominants. She's got 2 options down here. She can have another of the dominant, or she can have the recessive. She will see the full head of hair because she already has one healthy one. Let's assume she is heterozygous, which means she's going to have one healthy capital R and one lowercase r. In sex-linked traits, this is also called a carrier because she doesn't express the phenotype of the disease or the disorder, but she actually carries it secretly in her DNA. So then we're going to battleship all the little letters on top because we already have our X's and Y's built out. So now we put our green and our red R's where they're battleshipped up. Again, if you don't know how I built this, refer to the monohybrid cross. So there we go. We have 50% chance for boys, 50% chance for girls. Maybe the question wants to know, what is the chance my offspring are bald? Top left is a heterozygous female carrier. She's not going to be bald. That green R saves her from being bald. Top right, that's a capital R dominant, strong gene for hair, no bald male. Bottom right, unfortunately, that's going to be a bald dude. He's got the bald gene, and he has nothing else to balance it out. He's bald. Sorry. And unfortunately, on the bottom left, this female, also bald, because she is homozygous recessive. So there's a 50% chance I have a bald child. Be careful though. They can ask this question in a number of different ways. They can also say, what's the chance I have a bald boy? So I am going to still have this bald kid, but I'm really only going to look at the ones on the right. I'm not looking at the XX. So if it says, what's the chance I have a bald boy? It's still 50% because it's 1 out of the 2 boys. Same thing if it asks, what's the chance I have a bald girl? It's 1 out of the 2 on the left, so it's 50% chance to have a bald girl. So here's why you can kind of blame mom if you're bald. Say we change the mom's diseased R, get rid of it, and replace it with a capital dominant R for full head of hair. So now she has no male pattern baldness anywhere. What is that going to change on our Punnett Square? We're going to get rid of the 2 diseased ones, and our battleship looks very different. So now, what is the chance you have a bald kid? The answer is 0%. So, young men, if your dad is as bald as Bruce Willis, you are not necessarily going to get that, so do not freak out quite yet. Because if your mom has the double dominant, the homozygous dominant R, there's no way you're bald. And there's no way you're even a carrier, because you're a boy, and you can't be a carrier for a sex-linked traits. Now, the only thing about that is if you do have a bald dad, your bald dad is creating all these carrier women. Your bald dad is the reason that carrier female and that carrier female exist. So if they go on to find a husband of their own, they are now the carrier, and if he's bald, they're putting their kids in a bald predicament because they go back to the 50% from the last problem. So that's why people say baldness is not actually transmitted through the father, it's transmitted through the mother. And they're kind of right, because just because your dad's bald doesn't mean anything as long as your mom has 2 healthy sex-linked alleles, and her genotype is homozygous dominant. So a quick little note before we go. The hardest thing about this is figuring out what to put in the genotype portions. In my Punnett Square, I gotta figure out what goes here, 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 and here. And that can be kind of difficult sometimes. It's not always going to give you the exact genotypes or letters. It may not even have letters in the question at all. It's going to use a lot of vocabulary. It's going to use homozygous. It's going to say heterozygous, dominant, recessive. It's going to say this person has a phenotype of this. And you have to assume something else. So for example, I'm going to do a blood type problem that's kind of tricky. Say the mom is type A and dad is type O. Got the dad up here, the mom on the left. Because the mom is type A, we do not know what goes in the second part of her genotype. It could be another A, or it could be an I. It didn't specify, and without doing genetic testing on the mom, most people don't know that they are carrying anything else. So in a problem like this, maybe you gotta put a question mark, because you don't know. So all you can do is cross up what you know with the information given. And those are potential kids. So we could have heterozygous A, and we know we're for sure at least going to have a chance of this because it's in the top 2. The bottom ones could be more heterozygous A, or it could be I's. We don't know what the second one is. So one of the questions that I've given kids that is kind of tricky is, what if I tell you they have 3 sons? They have one son, let's say his name is Jack. He's type A blood. They have another son, his name is Bob. He is type O blood. And then their third son, Mark, is type AB blood. Using the information given, even if we don't know what that question mark is, can we tell which of these kids is not their biological son? Yes. It's got to be Mark. The reason is there's no way he could get that B from either parent. Even though we don't know what this question mark is, because we know the mom is type A, we know it can't be a B, or else she would have been type AB. It had to have been that IA or the little I, neither one of which is going to get us an AB for poor Mark. So maybe Mark is a half-brother from another marriage, or he's adopted, or whatever. But we know his blood type did not come from these two parents. And we'll hope that the mister knows this, because if he gets his son's blood tested for maybe a surgery or procedure or just to give blood, and his son comes back AB, and he thinks that's his biological son, well then mama has some explaining to do.